Epigenetic reprogramming. Can we actually make old cells younger? For decades, aging was treated as something we could slow down, but not fundamentally change. We can exercise, improve our diet, treat diseases, and monitor dozens of health indicators. But the basic problem remains: our cells change as we get older. Now, a field of biotechnology called epigenetic reprogramming is trying to approach that problem from a completely different direction. And in 2026, this research is moving from laboratory experiments toward early human clinical trials. So what exactly are scientists trying to do? Our DNA contains the instructions used by cells to function, but DNA is only part of the story. Cells also have a complex system of molecular signals that determines which genes are active and which ones are switched off. This layer of biological regulation is called epigenetics. As cells age, these regulatory patterns can change. The DNA sequence itself may remain largely intact, but the way the cell reads and uses that information can become less efficient. The idea behind epigenetic reprogramming is surprisingly simple in theory. What if scientists could reset some of these age-related changes and make an old cell behave more like a younger cell? The challenge is doing this without completely changing the identity of the cell. Researchers have been studying a group of proteins known as Yamanaka factors. These factors can dramatically change the state of a cell. But there is an obvious problem. If you completely reprogram a specialized cell, you can essentially erase its identity and push it toward a stem cell-like state. That is not what doctors want. The goal is therefore something much more controlled: partial reprogramming. Instead of completely resetting the cell, researchers are trying to push it part of the way toward a younger biological state while allowing it to remain a functional liver cell, nerve cell, retinal cell, or another specialized cell. This is where the technology becomes extremely complicated. The timing, dosage, combination of factors, and delivery method all matter. Too little reprogramming may have no meaningful effect. Too much could create serious biological problems. And that brings us to one of the most important developments in 2026. Biotech company Life Biosciences received FDA authorization to begin a phase 1 clinical trial of its therapy known as ER100. The treatment is designed around partial epigenetic reprogramming and initially targets diseases affecting the optic nerve. The company has reported that the first patient was dosed in June 2026. This does not mean scientists have discovered a treatment that makes people younger. Phase 1 trials are primarily about safety. Researchers need to determine whether the treatment can be delivered safely, whether patients tolerate it, and whether there are early signs that the biological approach is working. That distinction is extremely important. We are not talking about a proven anti-aging treatment. We are talking about an experimental technology entering an early stage of human testing. Another company attracting attention in this field is Limit. Its approach also focuses on epigenetic reprogramming, but the company is using artificial intelligence as part of the discovery process. The problem is scale. There are enormous numbers of possible combinations of biological factors that could potentially influence cellular behavior. Testing all of them manually would be incredibly slow. Machine learning systems can help researchers analyze biological data, identify promising targets, and prioritize combinations for laboratory experiments. Limit says its AI-driven systems are helping accelerate this discovery process. And in June 2026, the company announced a $435 million funding round to continue developing its technology and move toward clinical research. This combination of AI and biotechnology could become one of the most important parts of the story. Instead of researchers manually searching through thousands of possibilities, AI can increasingly act as a filter between huge biological datasets and laboratory experiments. But there is still a very long road ahead. A result observed in a cell culture does not automatically translate to a treatment for a human being. A treatment that works in one type of cell may not work in another. And even if a cell becomes biologically younger, that does not automatically mean the entire body becomes younger or that a person will live longer. That is why the early clinical trials are so important. The researchers now have to answer a much harder question. Can partial epigenetic reprogramming produce a useful biological effect in humans without creating unacceptable risks? If the answer is yes, the implications could be significant. Instead of treating aging only as a collection of separate problems, medicine could potentially target some of the underlying cellular changes associated with aging. But that future has not arrived yet. For now, epigenetic reprogramming is an experimental technology, and 2026 represents an important transition point. The science is moving beyond laboratory demonstrations. The first human trials are beginning, and artificial intelligence is becoming part of the search for the right biological interventions. The big question is no longer simply whether scientists can change the biological age of a cell. The real question is whether they can do it safely, precisely, and consistently enough to produce a meaningful benefit for patients. The next few years should give us the first serious answers.